Hello everyone and welcome to my YouTube channel Perfect Lab. The question is, have you ever wondered why sterilizing culture media and maintaining sterility are so important in the laboratory? This is because even a tiny contaminant can affect experimental results. So in this video, I will demonstrate a simple experiment to show the importance of sterilizing media and maintaining aseptic conditions, and how contamination can influence microbial growth. So let's get started. To better understand our topic, we will have to perform a simple experiment that will help us to demonstrate the importance of sterilizing culture media and keeping this media sterile in microbial work. So to do this, we need 5 different setups as shown over here, with the first flask as shown here containing autoclaved nutrient broth with no E. coli inoculation, and the second flask or setup over here contains an autoclaved nutrient broth with no E. coli inoculation. The third flask here contains an autoclaved nutrient broth with E. coli inoculation, and the fourth flask over here contains autoclaved nutrient broth with E. coli inoculation, and the fifth flask over here contains autoclaved nutrient broth with no E. coli inoculation but left open to the atmosphere as shown here. And in performing this simple experiment, what we have to note is that the incubation temperature is at room temperature, and the incubation location will be on the laboratory bench, and the incubation duration is 18 to 24 hours, but in some cases 24 to 48 hours as shown here. Observable results after incubating each setup of flask at the room temperature on the laboratory bench for 24 to 48 hours. So flask 1, which contains autoclaved nutrient broth with no organism or E. coli inoculation, shows no turbidity or the formation of sediments at the base of the flask to indicate that there is no visible microbial growth in the medium. And in this situation, the medium still remains clear as shown here. The flask 2, containing an autoclaved nutrient broth with no organism or E. coli inoculation, became turbid due to the growth of microorganisms already present in the unsterilized medium. And the flask 3, which also contains an autoclaved nutrient broth but inoculated with E. coli, became highly turbid with sediments formed at the base or bottom of the flask after incubation. And this observation indicates growth from both the inoculated E. coli and contaminating microorganisms in the unsterilized medium. Flask 4, which also contains autoclaved nutrient broth with E. coli inoculation, became uniformly turbid as shown here after incubation. And this is due to the abundant growth of the inoculated E. coli. And in some cases, the medium may have slight sediments after standing. And the flask 5, which also contains autoclaved nutrient broth with no organism or E. coli inoculation but left open to the atmosphere in the laboratory, became slightly cloudy. And this cloudiness may be as a result of contamination from airborne microorganisms that would have entered the flask whilst it was left open. So in summary, these observations clearly demonstrate that 1. Sterilizing or sterilization prevents unwanted microbial growth as shown in flask 1, and 2. Lack of sterilization allows contaminants to grow which is also shown in flask 2. And 3, failure to sterilize your prepared medium before inoculation results in mixed microbial growth as shown in flask 3. And 4, sterile medium with proper inoculation supports growth of only the intended organisms as shown in flask 4. And 5, exposure of sterile medium to air leads to environmental contamination as shown in flask 5. So let's try and look at some questions with their answers for an in-depth or better understanding of the simple experiment that we just discussed. So the question is, based on the experimental result, what conclusions can you draw about the importance of 1, sterilizing culture media, and 2, maintaining sterile cultures, and 3, factors that lead to bacterial contamination? So these are the answers. I will just leave it for you to go through. So thanks for watching, and if you found this video helpful, please like, share, or subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.